Now we go to explain the second mechanism how to coagulate casein, and this one is by enzymes, and this occur in making cheese. The other one is in yogurt, this one is mostly in cheese. Kappa casein, the one on the surface of casein cell, has 169 amino acids, and then you remember the casein micelle and part of the kappa casein are extruded outside like this, the other part is still inside. And then the enzymes, the rennet enzyme, the one that we use in making cheese, will just hydrolyze at this position. We'll split this one. We'll cut this part away. When it's cut this part away means that the remaining casein micelle become hydrophobic because the hydrophilic parts now are removed. So now the micelle, the remaining micelle become hydrophobic. Hydrophobic, then they don't want to have a contact with water, so they come together to lower the surface area to have less contact with water. And then the result is the coagulation. So here you have kappa casein, and then the enzyme will just split this. This part is hydrophilic from 106 to 169 amino acid. Hydrophilus and is soluble is washed out in the way when we make cheese, and the remaining part of the kappa casein, the one inside here, still stay inside the micelles. Okay, and so by enzyme, the coagulation is actually based on hydrophobic interaction. This is the term we call hydrophobic interaction. Right? That's the main mechanism in making cheese. And then in chapter 7, we will discuss making yogurt, we will discuss making cheese, and we will see again mechanism. Do you have question for this part? Is it already clear for you for the coagulation of caseins?
Yes, sir.
Okay. When you work in the lab, when you do tests, for example, you normally use the other methods. Maybe you use alcohol. Alcohol can also coagulate proteins, especially alcohol at high percentage. For example, it's the high purity, you can coagulate proteins. The other method people can coagulate protein is to use heat. If you heat intensively enough, then the protein will be denatured and will form a gel or will coagulate. This is another method. And then I think like this one, then you use more in the lab by heating, by acid, by salt, multivalent salt, can be calcium or magnesium and so on, or be enzyme in making cheese. These are more industrial application.